everyone, welcome back to Bloom with Bio. So today we are going to discuss the next chapter, that is excretory products under elimination. As you all know, our human body constantly undergoes a lot of metabolic activities, and as a result of all these metabolic activities, nitrogenous waste are produced in our body, and these nitrogenous waste has to be removed from our body to maintain the homeostasis and also to prevent our body from toxicity. And the process of elimination of this nitrogenous waste from our body is called as the excretion. So today in this chapter we will be learning about these different types of nitrogenous waste, human excretory system, nephrons, the structure of nephrons, mechanism of urine formation, regulation of their excretion, disorders related to excretory system. So we'll move on to the chapter now. So first we are going to learn what are the different types of excretory products. The most toxic type of excretory product is ammonia. And the organisms who remove this ammonia as their excretory product is called as the ammonotelic organisms. And this ammonia, since it is very toxic, it requires a large amount of water for its elimination. So the animals who eliminate ammonia as their excretory product is aquatic organisms. And the organisms who remove ammonia as their excretory products will remove it through their body surface as well as through gills as ammonium ions. So examples are aquatic insects, then aquatic amphibians, bony fishes, and tadpole. These tadpoles are nothing but the larvae of frog, that is an amphibian. Coming to the next type of nitrogenous waste, that is the urea. Comparing to that of ammonia, urea is comparatively less toxic, and this is having a moderate toxicity, so it requires comparatively less amount of water for its elimination. So comparing to ammonia and urea, ammonia is 1 lakh times more toxic than urea. So you can compare the toxicity of ammonia and urea now, right? And the organisms require less water for its elimination. And the terrestrial organisms will have urea as their excretory products. And these organisms are called as ureotelic organisms. And example, we have terrestrial amphibians, then cartilaginous fishes, mammals, etc. So the next type of excretory product is the uric acid, and this is the least toxic one. Okay. So the organisms who remove uric acid as their excretory product is called as the uricotelic organisms. So they are uricotelic organisms. So examples are terrestrial insects, birds, reptiles, etc. The excretory organs of different animals. So first one is platyhelminthes. They have the excretory organ, that is the flame cells. Next we will see organisms like earthworms. So these earthworms have nephridia for their excretion. Then crustaceans like prawns, they have green gland, also called as the antennal glands, for excretion. Insects have Malpighian tubules for excretion. Then coming to amphibians, reptiles, aves, and mammals. They all have kidney as their excretory organ. Now coming on to the human excretory system. So human excretory system consists of a pair of kidney, then they have a pair of ureters, a urinary bladder, and a urethra. So pair of kidneys, pair of ureters, a single urinary bladder and a single urethra. And above these kidneys we have the adrenal glands present. So kidneys are dark brown bean-shaped structures, and they are located in the lower abdominal cavity on either sides of the vertebral column. And these kidneys are the place where filtration will occur. Now coming to the ureters. So whatever filtrate was produced in the kidney will be taken towards the urinary bladder through these ureters. Urinary bladder is a sac-like muscular organ which will store the urine until it is eliminated. And urethra is the passage through which the urine is eliminated outside from the urinary bladder. Now, in male and females, urethra is a little bit different. Females have a short urethra that is like 2 to 3 centimeters, and males are having a long urethra that is up to 15 to 20 centimeters. Why urethra in males are long? These urethras act as the common passage for urine as well as the sperm. So this urethra in male is also called as the urinogenital tract. And now we are going to see the structure of kidneys. Kidneys are covered with a renal capsule, and this renal capsule have an outermost layer that is the renal fascia, and inner to that is the adipose tissue, which acts as the protection, or which provides protection or acts as a shock absorber. Then inner to that, the kidneys are divided into two regions. The outer region is called as the renal cortex, and the inner region is called as the renal medulla. So in the renal medulla, there are pyramid-like structure or triangular structures called as the medullary pyramids, or it's also called as the renal pyramids. And these pyramids are pointing into the renal calyx. Okay. And the region which extends into the renal calyx in between the medullary pyramids are called as the column of Bertani. And the region through which the urethra comes out and blood vessels and nerves will enter into the kidneys are called as, called as the hilum. And the next one is nephrons. These nephrons are also called as uriferous tubules, and these are the structural and functional unit of kidney, and there are 1 million nephrons in each kidneys. These nephrons have two parts. One is the glomerulus and the other one is the kidney tubules or renal tubules. Renal tubules includes the Bowman's capsule, PCT, DCT, loop of Henle and collecting duct. Now we will see the structure of nephrons and what all are present, or how we can draw the structure of nephrons. Coming to the structure of nephrons, we have the tuft of capillaries, that is the glomerulus, the Bowman's capsule, which is the cup-like structure which contains this glomerulus. Then we have the first highly coiled structure, that is the proximal convoluted tubule. Then we have loop of Henle, one descending loop of Henle and the other one is ascending loop of Henle. Then we have DCT, that is distal convoluted tubule. Then the collecting duct is present. And now blood is taken into the nephrons by the afferent arterioles, and the blood is taken away from the nephrons by the efferent arterioles. So now you can see here that the glomerulus, Bowman's capsule, PCT and DCT are lying in the cortical region of the kidney. Collecting duct and loop of Henle is located in the medullary region of kidneys. So now we are going to see the functions of kidney tubules. So first we will see the functions of PCT. PCT is made up of this is the region of PCT, and this is made up of brush bordered cuboidal epithelium. And PCT helps in maximum reabsorption of water. So whatever filtrate is filtered from the glomerular filtrate into the Bowman's capsule, this filtrate passes into the proximal convoluted tubule, and maximum reabsorption of substances will take place in this region. So you can see here 65% of water is getting reabsorbed in the proximal convoluted tubule. Then the ammonia, glucose and all, the complete ammonia and glucose is getting reabsorbed in PCT, and 90% of bicarbonate is getting reabsorbed, and sodium and potassium 65% is getting reabsorbed in the PCT. And this is the region where maximum reabsorption of substances is taking place. And the next one is loop of Henle. So this is the descending loop of Henle, and this is the ascending loop of Henle. So descending loop of Henle is impermeable to electrolyte and it is permeable to water. So this allows the movement of water outwards. So you can see here, this allows the movement of water outwards, and the electrolytes will be remaining inside the loop of Henle. So what happens as we move down to the medullary interstitium or medullary region, the concentration will increase. So at this point, the filtrate will be the maximum concentration. Now moving on to the ascending part of loop of Henle, what will happen? The ascending part of loop of Henle is permeable to electrolytes but impermeable to water. So now water cannot move out, but instead the electrolytes will move out. So this leads to the reduction of concentration of filtrate in the ascending loop of Henle. And from the ascending loop of Henle, the filtrate is given into the distal convoluted tubule or the DCT. and DCT is the place where conditional reabsorption of water will take place. So here water will take place depending upon the condition. That is why it is called as conditional reabsorption. If water is there more in our body, if I am drinking a lot of water, there will be sufficient amount of water in my body. So a lot of reabsorption will not take place. But if in a condition where I am not drinking a lot of water, if I am not drinking water properly, so what will happen at this time? My body requires water, right? So whatever filtrate is coming to DCT, more amount of water will be reabsorbed here, so that to maintain the blood volume. Now understood what is conditional reabsorption of water. And sodium, potassium and ammonia are also secreted into this DCT to maintain the osmotic potential. And also like uric acid, organic acids or ammonia are also secreted into the proximal convoluted tubule. Why are these substances secreted into the PCT and DCT? And this is to maintain the osmotic balance of our body. Electrolytes, water and urea will be getting reabsorbed, and whatever urea is getting reabsorbed will go to the medullary interstitium, and it will help in the maintenance of osmotic balance in the medullary interstitium. So sodium and urea are the two substances which helps in maintaining the osmolarity in the medullary interstitium. So what is medullary interstitium? It is a region in the medullary region which is surrounding the loop of Henle and the conducting duct. So the osmotic concentration or the osmolarity of this region is maintained by the urea and the sodium. There are two types of nephrons. One is cortical nephron and the other one is juxtamedullary nephrons. So in our body, most of the nephrons are cortical nephrons. That is 85% of nephrons in our body is cortical nephrons, and just 50% of them are juxtamedullary nephrons. Cortical nephrons are mainly found in the cortical region, and juxtamedullary nephrons are mainly found in the medullary region. So cortical nephrons have a very short loop of Henle and in the cortical region, and juxtamedullary nephrons have long loop of Henle, and that is in the medullary region. So in this cortical nephrons, vasa recta is not present, and in juxtamedullary nephrons, vasa recta is present. So what is vasa recta? These are minute networks of capillaries which are found around the loop of Henle, and this helps in reabsorption of water as well as helps in maintaining the osmotic balance in our kidneys. And these vasa rectas are running parallel to that of your loop of Henle. So next we are going to see the mechanism of urine formation, and this includes three steps. So the first one is ultrafiltration, then reabsorption, and the secretion. So the reabsorption is occurring from the tubule to blood, and whereas the secretion is occurring from the blood to tissues or tubules. So now we are going to see all these steps in detail. So now we are going to see what is ultrafiltration. So approximately about 1100 to 1200 ml of filtrate is produced by our kidney in 1 minute. So this is like approximately 1/5 of the blood which is pumped out from each ventricles in a minute. So now we are going to learn what is ultrafiltration. So why it is called as ultrafiltration? Because when the glomerulus is filtering the blood into the Bowman's capsule, all the things like glucose, amino acids, bicarbonates, water, electrolytes, waste, everything is produced or everything is taken into the Bowman's capsule. Now from there, from proximal convoluted tubule, distal convoluted tubule and the loop of Henle, everything has to be reabsorbed back into our body. So approximately 1100 to 1200 ml of blood is filtered out by each kidneys per minute. And this is about like 1/5 of the blood which is pumped out by each ventricles in a minute. So actually for this filtration or this diffusion to happen, 3 layers should be there. Like we have learned the diffusion membrane in alveoli and between the alveoli and blood vessels. Same diffusion membrane is present between the glomerulus and the blood vessels. So we will see what it is. So we have the first one as the endothelium of glomerulus. So now we have the endothelium of glomerulus. Then we have the basement membrane and the Bowman's capsule's epithelium. Epithelium of Bowman's capsule. So the epithelium of this Bowman's capsule is called as the podocytes. And these podocytes have small minute pores called as the slit pores. and through these only the ultrafiltration is taking place. And the amount of filtrate that is produced by the kidney is called as the glomerular filtrate. And the glomerular filtration rate is like 125 ml per minute, and it is approximately equal to 180 liters per day. But still we remove only 1.5 to 2 liters of urine per day. So what does that mean? So whatever filtrate is, glomerular filtrate is produced, more than 90% of that is getting reabsorbed into our body back. So our kidneys have a built-in mechanism to regulate all these activities, and that is the juxtamedullary apparatus. And this is formed by the modification of DCT as well as the efferent arterioles. So when there is a fall in this GFR rate, what will happen? This juxtamedullary cells will produce renin, and this will stimulate the glomerular blood flow and thus bringing it back to normal. Next is reabsorption, which is done by the kidney tubules. So there are two types of absorption here. One is active reabsorption and the other one is passive reabsorption. So what is actively taken back and what is passively taken back? So actively, glucose, amino acids and sodium or electrolytes are taken back actively, and nitrogenous waste are taken passively. Let us consider PCT. In PCT, the reabsorption of water is passive. But coming to that of the loop of Henle as well as in collecting duct, the reabsorption of water is active. So next one is secretion. So secretion is done to maintain the osmotic balance of our body, and what all are secreted is that H plus is secreted, K plus is secreted, then ammonia is secreted. So these substances are secreted into the tubule so that it can maintain the osmolarity or osmotic balance in our body. So next we are going to learn the mechanism of concentration of filtrate. And according to most of you, I guess this is the most difficult topic of this chapter, right? But it is not at all difficult. This is the most important topic, and for NEET you can get full marks from this topic if you learn this properly. You can get a question sure and you can write the answer also. So now we will draw the loop of Henle and vasa recta here. So this is the medullary region. So the loop of Henle as well as the vasa recta is found in the medullary region along with your collecting duct. So this is the portion which differentiates the cortex and the medulla. Okay, you can see here the cortex and the medulla. So in the cortical region and the beginning of medullary region will have almost same osmolarity, that is approximately equal to 300. Okay, so moving down towards the medullary region, the concentration will increase, that is like 300, 400, 600, 800, 900 and 1200. So in the lowest portion of the medullary interstitium or medullary region, you have the highest concentration, and in the cortical region, near to the cortical region, what you will have? It will be having the comparatively lower osmotic pressure or osmotic concentration. So once the filtrate is reaching here in the descending loop of Henle, the blood will be moving in the downward direction. And once this reaches the ascending loop of Henle, what happens? The blood started moving in the opposite direction, that is upwards. So in descending loop it is coming downwards, ascending loop it is coming upwards. So that is why it is called as counter current mechanism. Understood what is counter current? So this is called as counter current. Again you can see the vasa recta in the, so this is the descending limb of vasa recta and this is the ascending limb of vasa recta. So in descending limb of vasa recta, the blood will be flowing in the downward direction, and in the ascending limb of vasa recta also the blood will be moving in the upward direction. So here also what happens? The flow of blood is in the opposite direction. So it is called as counter current. So understood what is counter current? The blood and the filtrate is moving in opposite direction in both the limbs of vasa recta as well as the ascending limb and descending limb of Henle's. So that is why it is called as the counter current mechanism. So what happens here? This is the descending limb of loop of Henle and this is ascending limb of loop of Henle. So we have already learned that the descending limb of loop of Henle is permeable to water and impermeable to electrolytes. Okay, this much is clear. So once the water is taken away and the electrolyte is remaining in the loop of Henle, what will happen? The electrolyte concentration will increase. So it is like from 300 moving downwards, it will become like 400, then it will become 600, then it will become 900, and once it reaches the lowest portion, it will become 1200. Okay, this much clear. Now what happens when the filtrate is passing in the ascending limb of loop of Henle? This ascending limb is impermeable to electrolyte and impermeable to water. So electrolytes can freely move out, but water is not allowed to move out from here. So since water is staying there and electrolytes are moving out, so the osmotic concentration start decreasing here, like from here like 900, 800, 600, 400, 300. So once it come back to the cortical region, the concentration will come back to the normal. But what happens here, this filtration amount or the filtrate amount is reduced. Okay. So now this is taken into the DCT where conditional reabsorbing will take place. Now okay, that is none. Now whatever NaCl is released into the medullary interstitium is taken by the descending limb of vasa recta. So whatever electrolyte is released out by the ascending limb of loop of Henle is taken by the descending limb of vasa recta. Understood? And this is taken and released into the region near descending limb of loop of Henle. Same way, whatever water is given out by the descending limb of loop of Henle is taken up by the ascending limb of vasa recta. Now, once the filtrate reaches the collecting duct, so this is the collecting duct, what will happen? Water as well as urea and sodium chloride. Sodium will be reabsorbed. So, the urea which is reabsorbed will be given back into the medullary interstitium. Sodium which is also collected back will be taken back to the medullary interstitium. That is why urea and sodium are the two components which helps in maintaining the osmotic balance of medullary interstitium. So now we are going to see the regulation in kidney. So how the kidney functions are regulated, we are going to see now. So imagine the blood osmolarity decreased. Okay. So this will give signal to the osmotic receptors in hypothalamus. And this osmotic receptors in the hypothalamus will initiate or will give signals to the posterior pituitary. And this posterior pituitary will produce ADH, and that is the vasopressin. And this increases the permeability of DCT. And this leads to the reabsorption of water. And the osmoreceptors present in the hypothalamus will make us feel thirsty and it make us drink water. So the water which we drank and the water which got reabsorbed will help in decreases the osmolarity and thus maintains the blood osmotic balance or blood osmolarity level. So next we are going to learn renin-angiotensin mechanism. So here what happens when there is a fall in GFR, renin is produced by the JG cells. So juxtaglomerular cells will produce renin when there is a fall in GFR. And what will this renin do? It will convert the angiotensinogen to angiotensin 1 and then it will convert it again into angiotensin 2. And this angiotensin 2 will increase the glomerular blood pressure and thus by increases the GFR. So it also activates the adrenal cortex to produce aldosterone. And this causes the reabsorption of electrolyte and water. And this leads to the increase in blood pressure and the GFR. So thus the GFR and blood pressure is increased. So what happens when GFR and the blood pressure is increased? So the blood that is moving into the heart will increase. So the walls of heart will produce ANF, that is the atrial natriuretic factor. And this factor decrease the blood pressure as well as the glomerular filtration rate. So this ANF mechanism will check the renin-angiotensin mechanism. So now we are going to see what is micturition. It is a process of release of urine and this is controlled by a neural regulation that is called as the micturition reflex. So now in this diagram we are going to learn how micturition is taking place or how it is initiating micturition. So what happens, a urinary bladder, when it is getting filled with approximately 500 ml of urine, the structure receptors present in the urinary bladder will send signals to the central nervous system, that is the micturition center. So from the micturition center, another reflex is taken back into the urinary bladder, and that leads to the contraction of urinary bladder muscles, as well as the sphincters in the urethra will be opened, and thus lead to the elimination of urine called as the micturition. So now we are going to see excretion by other organs. So first is lungs. So from lungs, what is getting eliminated is the carbon dioxide, which is the byproduct of the metabolic activities. Now from the liver, what is getting eliminated? That is the bile pigments, then the cholesterol, then the degraded steroids, then hormones and vitamins. These are, whatever is not required, these are removed by the liver, and that is through the digestive system. Now coming to skin. Skin contains two types of gland. One is sweat gland and the other one is sebaceous gland. So the removal of sodium, water, lactic acid and urea is done by the sweat glands, and the elimination of steroids, hydrocarbons and waxes is done by the sebaceous glands. Now next we are going to see different disorders related to the excretory system. So first one is uremia. So this is accumulation of excess urea in blood. So when the amount of urea in blood is more, it is called as the uremia, and this can be removed using hemodialysis. So what is hemodialysis? It is artificial kidneys. That is, blood is taken out from the patient's body, passed out into a dialyzer, and from there it is taken back into the human body after filtration. So once the blood is taken out for filtration, it is mixed with the heparin, which is an anticoagulant, so that the blood won't clot. So once it is taken back into the human body, or once it is taken back into the patient's body, it is treated with the antiheparin so that the blood will clot if any wounds are there. Next is the renal failure. This can be due to any accident, injury or any serious illness. Then comes the renal calculi. So these are the stones or any insoluble crystalline salts which are accumulating in the kidney. And the last one is glomerulonephritis. This is nothing but the inflammation of the glomeruli. So that's all with the chapter. So we have learned types of excretory waste, different organs present in different organisms for excretion. Then we have learned human excretory system, nephron structure, mechanism of urine formation, regulation of kidneys, as well as disorders related to the kidneys or excretory system. So I think you have understood this. So if you have any doubt, you can drop a message in the comment section, or you can directly send me messages. I'll be clearing it for you. So thank you and have a nice day.